Yes, everybody, very good evening all and very warm welcome to the session. So happy to meet you all back in this awesome session. So this is myself, Dr. Saranya. We are listening to the revision concepts for your CSIR NET. Yes, so if at all you have not yet visited us at Unacademy, please do visit us. You can download the app and you can seamlessly attend so many sessions. So today we are going to see RNA polymerase. This is going to be the lesson 3. So already we have taken 2 lessons under this rapid revision topic. Yes, so this lesson number 1, it was on enzymes. Lesson number 2 we did with the microbial kinetics. So if at all you didn't see it yet, it's high time that you have to look at those rapid revision concepts. Yeah, and today we are going to see a next important thing. So that is going to be on the RNA polymerase. RNA polymerase are the key enzymes that participate in your transcription. Yes, so transcription, it is the process where it is going to convert your DNA to form the RNA. Yes, so DNA, it is getting transcribed to form your RNA. Right, so this is taken care of with the help of your polymerase. So this process is an integral part of your central dogma of molecular biology. So this takes place in both your prokaryotes as well as in your eukaryotes. But even though the basic mechanism is going to remain the same, there are minor differences between the prokaryotes and eukaryotes. Right, so this is a little bit about myself. If at all you have not known me so long, so I am glad to introduce myself. I am Dr. Saranya. I have completed my Ph.D. in Biomaterials after cracking the examinations. So I do have teaching and research experience. Right, so this is something about our Unacademy Plus and Iconic subscription. So in the Plus subscription, you will get access to the structured courses, and you can listen to them any number of times. So you can use it for revision and re-revision, everything you can use it. You will have weekly test, live classes, and in Iconic you will have a guidance, planning, analysis, and this is what is going to make you a little bit edge over the preparation. Alright, so if at all you have not yet subscribed, you can subscribe by using my code Saranya Live, so that will give you good discounts. Right, fine, great. So this is our Telegram channel. If at all you have doubts, you want the notes, you can ping me there. I'll help you with that. And these are the special classes recently conducted. So I have discussed the evening sheet paper, evolution MCQs, RNA editing. You can attend them for free. Yes, if you really want to, no money is really a constraint and you want it to attend for free, definitely you can take this. Yes. Okay, that's all folks. So let's begin with our RNA polymerase. Right, so this RNA polymerase, it is going to convert my DNA into this RNA. Okay, so the first point is that in case of replication, okay, so in case of replication, what is the primary enzyme? So the replication enzyme, it is going to be my DNA polymerase. Okay. So does it require a template? Yes or no? Yes, this is RNA polymerase. This is a transcription enzyme. This requires a template. My RNA polymerase, yeah, it also requires a template. So what should be the direction of the template? Here it is going to be 3' to 5'. What about my RNA polymerase? Same, it is going to be 3' to 5'. So what is the template nature? Template, it is going to be the DNA. So I can call this as DNA dependent DNA polymerase. This is going to be DNA dependent RNA polymerase. Please familiarize yourself with these phrases because this is usually asked up in your examination. DNA dependent DNA polymerase, RNA dependent DNA dependent DNA polymerase, and one more fellow you have. Do you remember who is this? RNA dependent DNA polymerase. What do you call them as? Do you remember what is this RNA dependent DNA polymerase? It is nothing but your reverse transcriptase, right? And also your telomerase. So these telomerase and reverse transcriptase are depending on the RNA template. Okay, so that is why you call them as RNA dependent DNA polymerase. Clear? Everybody in the class, did you get clarity into this? What is your DNA dependent, RNA dependent and all? Yes? Okay, fantastic. So if at all you are having any doubts, please feel free to ask me, such that I will be able to guide you better. Okay, right? That's fantastic. So now let us make up a move. So this is about my template. Okay. So as far as your DNA replication is considered, it is requiring a primer, right? A primer is mandate. So without primer, yes, it is not going to work. Correct? Yes. But what about my RNA polymerase? RNA polymerase, does it require a primer? Not at all. So no primer is going to be required. Got the point? Yes. So my DNA dependent DNA polymerase alone require a Shreya. Correct. So this alone requires a primer, but my RNA polymerase does not require a primer. Okay. So what is the direction of action? Okay. So my DNA polymerase, it will move in 5' to 3' OH. I mean 3' direction, because it is going to extend your 3' OH with your 5' phosphate. So this is going to be your phosphodiester linkage. Fine. So what is happening with my RNA polymerase? Do they move in the opposite direction, or what sort of direction it is going to move on with? Yes, this is also going to move only in 5' to 3' direction. Okay. So majority of your enzymes, this enzyme's mode of action, yes, it is going to be the same direction. Okay. But if you see the end product, in case of your replication, you have both strands, isn't it? So it will be 5' to 3', your leading strand, and the lagging strand, it is going to be in your 3' to 5'. Right? So both strands will be produced in case of your DNA, but in RNA only one strand. Got the point? What is the strand? It is only 5' to 3'. Okay? Yes, majority of the RNA it will be single stranded. DNA is always double stranded. Majority of the RNA it is going to be single stranded. So single stranded means it is in what direction? It is going to be only in 5' to 3'. Why? Because my polymerase it is in direction 5' to 3'. That is why this is also in the same direction of 5' to 3'. Got it? Clear? Right? So this is clear to you? Fantastic. So now you have two strands. So what is the template strand? Template strand is 3' to 5', and one more strand, it is 5' to 3'. Okay, right. So now you produce an RNA transcript. So this RNA transcript, it is exactly looking like your non-template strand. So this is what is called as the coding strand. Yes. Yes, Vishnu, good afternoon. So this non-template strand is what is called as the coding strand, because it has the sequence exactly similar to that of this. Okay? Right. Fine. So our RNA synthesis it is going to be catalyzed by means of the enzyme. So that is what is called as our RNA polymerase. Okay. So where should my RNA polymerase bind? Can it bind anywhere? No. It is binding to a specific region. So that is what is called as the promoter. Okay. So where my RNA polymerase is binding, it is binding to the promoter. Okay. So in prokaryotes, right? Yes. What is the common promoter? You call them as the Pribno box. So what is this Pribno box? Nothing but it looks like our Tata box, isn't it? Tata at. Where is the location? Where is the location? It is at -10. That is why this is also called as -10 box. The same thing if it is going to occur in the eukaryote. Okay, it is usually called as Tata box. Okay, and this is going to occur at -35. -35 means it is going to be upstream. Okay, so -35 is it is going to be upstream, right? So this is what you have to remember. This is the location of your promoter. So this promoter, it is going to be the region where your RNA polymerase can bind. Okay, right. So in case of prokaryotes, which region of the RNA polymerase will bind? It is going to be the sigma region. Okay, yes, sigma segment or the sigma fragment, whatever you call the sigma, it is the subunit that is going to bind to the promoter because it is having all the specificity. Which is having the specificity? Your sigma factor. Your sigma factor is only having the specificity, so that is only going to bind with the promoter. In case of eukaryotes, you have the transcription factors. Transcription factors are present in prokaryotes. Have you ever seen, have you ever read that your prokaryote is going to contain a transcription factor? Not at all, isn't it? Yes, yes, Niti. So these one, they do not contain any transcription factor because directly your sigma segment will bind. But in case of eukaryote, this is going to make use of transcription factor like your TBP, Tata box binding protein. So that is only going to make a binding with that of the promoter. Understood the difference between your prokaryotic and eukaryotic transcription? Everybody in the class, you gotten clarity? Good. Fine. So now coming back to the theory. So now the promoter, it is going to include the first base. So that is going to get transcribed. Right. So from that your RNA polymerase is happily moving down. Okay. So this is how your RNA polymerase looks like. Yes. Fine. So in case of replication, you have a helicase. Correct. Right. In case of prokaryote, do you have a separate helicase? In case of prokaryote, no. Your polymerase is only functioning like a helicase that is going to unwind. Okay. In eukaryotes, in transcription, you have a separate factor called TF2H. So that will do the unwinding. This is working like a helicase. Yes, Chiku. So this is how it is going to work like a helicase. But in case of your prokaryote, your RNA polymerase itself, it helps in your unwinding. Okay. So this is clear. I used to tell that whenever it is going to be some small shop. Okay, so visiting a small shop nearby. Okay, yes, the same person will be showing the goods to you. The same person will be billing. The same person will give the delivery to you. But if you go to the big shops, what happened? There will be few people who will be showing the addresses to you. Few people will pack, few people will bill. Finally, few of them will put the seal and give you the delivery. So, if it is going to be the higher model, they have separate individuals for every task. Okay, so for every task they are going to contain the separate individuals. Okay, but in case of smaller shops, yes, all in all, yes, we have only one person like all in all, he will be doing everything for you, right? So that is what it happens with the small shops. So that is what it takes place in your RNA polymerase also. In case of prokaryote, since because it is going to be a lower model, okay, so in this lower model what will happen? All the duties will be performed by the RNA polymerase. No separate helicase, no separate enzyme for your binding and all, everything the same fellow is doing. But in case of replication, you have a separate helicase, right? Likewise in eukaryote also you have a separate transcription factor like 2F2H, which will be carrying over all these unwinding activity. Clear? Right? Great. Fine. So what are the steps that is involved in your transcription? Right. The first step, it is going to be the initiation. So initiation. So major thing or step, it is going to be the promoter recognition. Okay. So once the promoter is recognized, then the bubble will be created. Once the bubble is going to be created, then what will happen? Your RNA synthesis will take place. Second one, it is called as elongation. So elongation is where the bubble is going to move. Okay, so then your RNA, it is going to be synthesized. Third one, it is going to be the termination. So termination means finally everything is over. So it is going to release. So in your prokaryotic as well as in this eukaryotic, in both of them, these three processes will definitely take place. Right. I told you in case of prokaryote, all in all it is conducted by a single individual. So in prokaryote there is only single type of RNA polymerase. Okay, you don't have a type 1, type 2, type 3 here. In prokaryote there is one and only one type of RNA polymerase. But in case of eukaryote, there you have RNA pol 1, yes, RNA pol 2 as well as RNA pol 3. Okay, so in case of your eukaryotes, you will be containing all these three different types of the polymerase. Okay, right. So now let us see in detail about your prokaryotic. So in prokaryotic, yes, in case of prokaryotic, the single RNA polymerase, it is having two subunits, two major. Okay, yes, one it is called as APO, or you call it as a core enzyme. Okay, right. Other one is going to be the subunit. This is what is called as sigma factor. The sigma factor is for the specificity. So what are coming under the core enzyme? It is going to include 2 alpha. Okay, and then beta prime, beta prime as well as omega. Okay, right. Sigma is separate because sigma is going to be the additional subunit, because that is only going to tell the promoter specificity. I will tell you why. Okay, shall we just see little bit in detail? Yes. Okay, so if I take my RNA polymerase, this RNA polymerase, it is going to be containing the core enzyme. So what are the component of the core enzyme? It is going to contain 2 alpha, beta prime, and then omega. Then who is coming? Specificity factor. So what is the specificity factor? The specificity factor is what is called as sigma. Together they are making my RNA polymerase. Okay. Now what is the charge of RNA? RNA is positively charged or negatively charged? RNA charge? Yes, everybody. What is the charge of RNA? RNA is negatively charged. Correct? Right. So this core enzyme, it is going to be positively charged. Yes. So now what happens? Usually, if the charges are going to be opposite, they will attract each other. So, non-specifically, who will bind? Non-specifically, the sigma factor, it is going to bind. Non-specifically, the sigma factor is going to bind your DNA. Oh my god, I write it as RNA. It is DNA. To the DNA only it is binding. No? RNA it is not yet produced. Okay. So, non-specifically, the sigma factor is going to bind to the DNA. Okay. So, it is allowed. No. You want them to specifically bind. So, only you have the sigma factor. So, the sigma factor gives the specificity. So, throughout the DNA, it cannot bind anywhere as it please, but it has to bind only to the promoter region. Because this promoter region, it is going to be the recognition site for the sigma. But once it is initiated, after initiation, during the elongation, can it bind everywhere? During elongation, can my RNA polymerase bind everywhere? My initiation is over. See, this is my promoter region. Till here, I have my genes. This is going to be my +1. For instance, I am telling you, this is going to be +1. This is -10. This is like 200. So, this is the total length of my mRNA. Okay. So, this is going to be my DNA. This is going to be my mRNA. Okay. So, initially, this recognition is done by sigma. After the initiation is over, sigma will leave the situation. Okay. Right. So, now this is going to be my elongation. In my elongation, there is no sigma. So, if sigma is not there, okay, so my core enzyme, can it bind to this DNA or not? Yes, it will bind. Then only it should produce your mRNA, no, or else only promoter will be transferred. So, at this time, sigma will be exiting. Yes, this process of sigma factor exiting. Yes, Ragnar, thank you. So, thanks a lot, Ragnar. So, as the sigma factor is exiting, this step, it is what is called as abortive initiation, because now no more continuation of the sigma factor. Yes, so the sigma factor is going to move away. Got the point? So these are the key terms which you should never forget. Shall we just randomly check our memory? Everybody in the class? Yes? Shall we check our memory? All of you are ready for fast checkup? Thank you everybody for your things. Right. Yes. So, during my elongation, okay, right, sigma factor, it is disassociated. Okay, sigma factor is disassociated from the core enzyme. True or false? Very good. Yes. So, during the elongation, you don't require the sigma factor. Okay. So, sigma factor will be disassociated. No more you require the sigma factor. Right. So, the second one. Yes. Absolutely. So, my alpha subunit recognizes promoter sequence and binds specifically to the promoter. True or false? Come on everybody, think and answer me. My alpha subunit, it is going to recognize the promoter sequence and it is going to bind to the promoter. True or false? Is it alpha or sigma? It is going to be only sigma. So this is a false statement, isn't it? Only sigma subunit. It is for promoter binding. Okay. This equivalent is only going to be the TBP in eukaryote. Got it? Yes? Is this now clear to everybody in the class? Great. So what is the role of alpha subunit? This alpha subunit, it helps in the binding of activator. Okay, so it is helping in the binding of activator, and yes, so it helps in the binding of activator. Okay, apart from that, it is going to help in the enzyme assembly. Okay, so enzyme assembly is also taking place. Activator is also binding. This beta and beta prime, this is only going to form the catalytic center. This is only going to help in the catalysis. Okay, right. And your sigma factor, it is only for the promoter. Okay, so sigma factor is only for the promoter. So now all the different parts of your polymerase is thorough to you. Everybody in the class, you got a total idea of different polymerases subunit. This is exclusively for your prokaryotes. So see, if it is going to be the core enzyme, it can bind to any DNA. So core enzyme means it includes two of your alpha, beta prime as well as your omega. Yes, but if the sigma is there, it is reducing the non-specific binding. So it makes the holo enzyme to specifically bind to that of the promoter. So this is what is the key factor in the initiation. Now we are going to see what are the names of your sigma factor. I think several examination this question has come, right? Yes. Sigma 70. First fellow. Sigma 70. The sigma 70, this is only going to make the housekeeping genes. So sigma 70 is only going to make up the housekeeping genes. Yes, sigma 70, it is going to be for the housekeeping genes. Yes, the next one, sigma S. Okay, it is for the stationary phase and some stress response. So who is heat shock? Heat shock is going to be very good, RPOH, or it is sigma 32. 32 is only for the heat shock. Shall we just repeat it one more time? Everybody in the class? Yes? Okay, first one is sigma 70. Sigma 70, it is meant for the housekeeping genes. Sigma 32, this is only for the heat shock protein. Sigma S, this is for stationary as well as the stress response. Okay, right. So this is going to be RPOH and that fellow is RPOD. Okay, so RPOD is housekeeping. S is going to be stationary and stress response, and 32 is heat shock. What about sigma E? Sigma E, it is for your extracellular protein. Sigma E, it is for extracellular protein. Okay, right. Then sigma 54 for nitrogen assimilation. Sigma 54, it is for the nitrogen assimilation. Sigma 54 is there now. So that is going to be for the nitrogen assimilation. Okay, yes. Sigma F for flagella. Sigma FeCl for the iron metabolism. Iron metabolism. Yes, everybody? Shall we try to recollect it one more time? Yes or no? Come on. Sigma 70, sigma S, sigma 32. What else we learnt? sigma 54. What else did you remember? Come on guys, what else did you remember? Very good everybody, all of you are very bright. Good everyone, continue on. Yes, right? All right, fantastic. Good. Yes, all, absolutely. Right, and then your sigma 54 visa, sigma E visa, right? Yes, and then sigma F visa, and then sigma FECL. Yes, very good all. Sigma 70, this is going to be for your housekeeping. Sigma S, it is for stationery as well as few stress response. Then sigma 32, it is for HSP, heat shock. 54, it is for nitrogen assimilation. Right. Sigma E, E, it is for extracellular proteins. F for flagella. FECL for ion transport. Got it everyone? So you got an overall idea of various kinds of sigma factors. Yes, is it all clear now? So no more doubt and worries on the sigma factor. Right, fine. So now it's high time that we are going to make up a move towards the next one. It is going to be the eukaryotic. Okay, right, fine. So after initiation, that is going to be the elongation as well as the termination. Yes, right. So elongation is going to continue, and the termination, what are the two methods of termination in case of prokaryotes? You have a, so how is your termination taking place in prokaryotes? Yes, everybody, do you remember any point regarding the termination in your prokaryote? Yes. So how do your prokaryote help in termination? So it is going to continue. Your polymerase, it is going to continue on till it is going to reach the end. Correct. So when it is reaching the end, what it will do? So it is going to help to come off. So that is going to be your row dependent termination as well as the row independent termination. So these two are the important termination. Okay, so this row independent, it is going to your U, poly U and all, it is going to come in that thing only. Okay, right. So that is going to be the termination. So once the termination is done, right, then it is going to make up. Your RNA polymerase will be coming out. Okay, and then it is going to move on. All right. So now shall we make up a move towards our eukaryotic transcription? Yes. So in eukaryotic transcription, there are three types of RNA. Number one, this RNA polymerase 1. It is going to transcribe your 18S or 28S rRNA. 18S or 28S rRNA. Your RNA polymerase 2 will transcribe your mRNA. Right. Your RNA polymerase 3 will transcribe tRNA, 5S rRNA and small RNAs. Okay. This is a very important slide. Yes. So one more time shall we revise and see. Your RNA in case of your eukaryote. Okay. In case of your eukaryote, you have the RNA polymerase 1. You have the RNA polymerase 2, you have the RNA polymerase 3. Okay, so RNA polymerase 1, it is for your 5.8S and 28S rRNA. Okay, Pol 2, it is meant for your mRNA as well as all your small RNAs. Right. Pol 3, it is for your 5S rRNA, tRNA and then few small RNAs. Okay, right. This one point. One more, it is the alpha amanitin sensitivity. Okay. So what is this alpha amanitin? this is going to be an antibiotic. So this antibiotic is isolated from a mushroom. Okay, right. So this is showing the sensitivity. So it is in this order. Pol 2 will show greater sensitivity, and then Pol 3. Finally, Pol 1. Pol 1 is least sensitive to alpha amanitin. This is also another important question that is routinely asked in your examination. Got it? Yes? So these two are the important things regarding your eukaryotic things. Got it? Right. Fantastic, everybody. Fine. So now, as I told you, in our eukaryotes, you have a special requirement. What is that? It is the transcription factor. Okay. So can you guess what RNA is this? Pol 1, Pol 2 or Pol 3? This is going to be for your Pol 1. Okay. So Pol 1 is going to produce your 5.8S rRNA and 28S rRNA. Okay. So this is what is going to be your Pol 1. Okay. Got it? Fine. Yes, everybody. So let us see it in detail. So here what happens? There is going to be a transcription factor called as SL1. Okay. What is the transcription factor? Called as SL1. Okay. So this SL1, it is going to contain your TBP. What is this TBP? Tata bonds binding protein. So these three regions are going to be your SL1 domain, and one of them it is going to be the TBP. Together they will be binding to the promoter region. Okay. So now you see that there is an upstream element. Okay, it is very big. So this usually contains your enhancer. So the enhancer should be typically near your promoter. No. If it is far away, then it is going to make no meaning out of it. So what happens? One more factor called UBF. Yes. So it is going to be UBF. UBF is going to be another important factor. It is going to bind to the upstream promoter element. Okay. Right. So this is also a dimer. Yes. So shall we summarize? So these are the transcription factors for Pol 1. Okay, so usually you will study the transcription factor only for Pol 2. But remember, Pol 1 also makes or contains the transcription factor. Pol 1, it is going to synthesize your rRNA generally. So it is going to be your 5.8S rRNA as well as your 28S rRNA. Okay, right. So it contains a segment called UBF. It is going to be a dimer. It is going to bind with your upstream promoter element. Second, it is an SL1. It is basically a tetramer. So out of this tetramer, your TBP, it is going to be one of the components. Okay, it is a part of this tetramer. Okay, this is only going to bind to your core promoter. These two things are thorough or not? Yes. After this only your RNA polymerase will be coming and binding. Okay. So now you understood how it is coming and binding. So Pol 1 is thorough to everybody in this class. Yes. Okay. So the next one we are going to see, it is going to be your Pol 2. Pol 2 is a very interesting fellow. Yes. So Pol 2 transcription factor, it is going to occur in this order: D, A, B, F, E, H. D, A, B, F, E, H and then J. Okay. DAB, FEC, J. Okay. D, A, B, F, E, H and then J. First one is going to be D. So what is this D doing? This D, it is going to contain your TBP. Okay, so this component of this fellow, it is only going to be your TBP, and that is going to bind to your promoter. Okay, right. Second one is going to be the A. So what this A is doing? it will bind upstream to that of the Tata box. So upstream means usually it is the enhancer. Okay, so usually A, it is binding with the enhancer. This TF2B, it binds downstream to that of the Tata box. Okay, so it is going to bind downstream to that of the Tata box. Okay, so TF2D is only binding with the Tata box. You can imagine this is TF2D. It is binding with the Tata box. Okay, so some fellow is binding upstream. Who is binding upstream to the Tata box? It is going to be the TF2A. Okay, it is upstream. That is why you call it as an enhancer. Few fellows, it is binding downstream to the Tata box. It is going to be TF2B. Okay, so this only makes your RNA polymerase to come closer to your promoter. Okay, right, fine. The next one, DAB is done. The next one is FEH. So let us see this F. This F, it is going to accompany your polymerase as it is going to bind with the promoter. It is an accompanying person. It is also recruiting your TF2E. E, it is for promoter clearance, and H it is for H is functioning like a helicase. It is going to help in your unwinding and all. So it is phosphorylating your Pol 2. Yes, and your J, it is for your promoter clearance as well as the elongation. So DAB, FEH, J. These are the key factors, that is transcription factors, which is involved in your polymerase 2. Yes, so for your Pol 1, only 2. One is UBF that binds to UPE. The second one, it is SL1 which contains your TBP. That is only binding to the Tata box. That's all. But see, for polymerase 2, you have these many fellows. Yes, to make the functioning happen. See there, this is going to be the start point. This is going to be your TF2D. Within the TF2D, the TBP is present. Yes, and your RNA polymerase will come and bind with your Tata box, and it is going to make up the starch. Okay, so is it all clear now? Shall we make up a move towards your Pol 3? Okay, so now we are going to making a move for Pol 3. Pol 3, it is of 3 types. One, it is for your 5S rRNA. Other one, it is going to be for your tRNA, and then few small RNAs. Yes, Suraj. So other small RNAs. So these are the 3 things where your Pol 3 is going to work. So if you take the type 1, it is going to contain the box AC. Okay. So in Pol 2, you have transcription factors 2A, 2B like that. Here it is 3. That's all. Okay. Right. So there you saw 2. Here we are going to see 3. So this is going to be the box A and B. Okay. So this is going to be for the box A and B. To them, your TF3C will bind. Okay. And this 3F3B, it is going to bind it to the promoter. So we saw that in case of your Pol 2 also, yes, TF2B is only binding with your Tata box, isn't it? Same. TF3B, it is also binding with your Tata box. Okay, right? Got it? Like your 2B, the 3B is also behaving. It is also binding with the Tata box. So here it is going to be AB. So that is only your C. Right? So then your polymerase is recruited. It is going to on. Likewise, if you take your box AC, so that is going to be your next type. That is type 1. So type 1, it is for rRNA. Right? 5S rRNA. And this fellow, it is for so type 2, it is for tRNA. So tRNA is only containing AB. So tRNA is AB box. So it contains your TF B and C. This fellow is AC box, which contains A, C and then B. Okay, so rRNA, it is going to contain the transcription factors A and then C and then B. Okay, right. But if it is going to be your tRNA, it contains only C and B. There is no A transcription factor for tRNA. Got it? It is a very important piece of information, right? So I hope that today you enjoyed learning the different types of polymerases. Yes. So we compared and contrast between our replication with the transcription, and then we compared this prokaryote versus eukaryote. Okay. To summarize, the prokaryote, it is having only one type. It will have 2 alpha, 1 beta, beta prime, omega, and then sigma. Sigma is the specificity factor. Under the sigma, we saw about this sigma 70, sigma 32, sigma 54, sigma S, sigma E, yes, sigma F, and then sigma FeCl. Right. Under the eukaryote, we saw about this pol 1, pol 2, pol 3. Yes. So pol 1, it is going to be synthesizing your rRNA. This is for your mRNA, and this is for your tRNA. Here you have this UBF and SL1. Right. Here you have the Tata box, transcription factors DAB, FEH, J. Right. tRNA, you have your transcription factor 3C and then transcription factor 3B in one set. In other set, you have this 3A and then 3C and then 3B. Okay, and then another one is Oct PSC Tata, to which your factors are going to bind. Yes, everybody, I hope that the total RNA polymerase is in front of your eyes. If you have any more doubts, please feel free to ask me. Right? Okay, so your CSIR NET is seriously very close. So if at all you want to make a systematic preparation, okay, then you have to take up the plus or this iconic subscription. You can definitely make use of my code Saranya Live. Okay, so if you really want to take the classes, so currently revision classes are going on along with your PYQ and MCQ series, so you can join and make it out. All right, so nice everybody. So thanks a lot. Subscribe our channel and like, comment, share, and click the bell button such that you will get all the notifications. Yes, I'll catch you soon tomorrow with more information. All the very best. Yes, take care till we meet. Tata, bye bye everybody.